pioneers in that field. And then later moved on to studying the properties of ion channels in lipid bilayers, notably complicated channels like the IP3 receptor. This was very important work. More recently, she's taken on other tasks studying polycystins and the neuronal calcium sensor 1, which is a very interesting protein. And Barbara, if you could switch to the next slide, please.
This one, you want the next
one?
Yeah,
that's where I should have been actually, which this shows her title. And then now the next slide, I want to just mention that Barbara is one of the most energetic people that I know. She's been a fantastic mentor to an advocate for young and old alike in our field. She has the energy to start a company, and she's a prodigious baker who manages to incorporate calcium into baking and many other aspects of her life. And we're just really pleased to have her here today. And she's going to be talking to us today about intracellular calcium and the prevention of chemotherapy-induced side effects. So just one fast reminder that everyone needs to remember to mute their microphone right now. And when you have questions, type them into the chat box. And at the end of the presentation, I will be giving the questions to Barbara for you. Okay, Barbara, take it away.
Okay, so as you know, this is my title. And as Aldebaran mentioned, I started a company, so I have to give you a disclosure that I'm the co-founder of Osmol Therapeutics, which is a company that's using NCS1 to design therapeutics. So this, of course, is not new to anybody in this audience, intracellular calcium signaling pathway, but you can see that I have a very pared down view of what's important. And so on the left here, we have the IP3 signaling pathway where we have, and you notice I don't even have agonists because I don't really study what the agonists are. We mostly care that you make IP3 and turn on the IP3 receptor. And of course, we have voltage-gated calcium channels, ryanodine receptors, polycystin 2, and the pumps to return the calcium back to the baseline. But ER and SR have high calcium. There's low calcium in the cytoplasm, high calcium in the extracellular fluid. And the idea is to maintain low intracellular calcium, but use changes in calcium to be a signaling molecule. Okay, and back in the 1970s, people thought of these membrane proteins sort of as vegetables sitting in a bilayer, all separated except for pairs that are pairs. And I think this is an unrealistic model. It's, of course, none of us probably remember anymore what it's like to be in a crowded bar, but I think a better model is a crowded bar on a Friday night where it's not whether you're going to be touching other people in the bar, but how close the contact is and how long it lasts. And to show that this is a more realistic model, I show one example, which is a clathrin-coated vesicle. And here all the proteins are on the membrane so close and so packed in that you can't even see the lipid. Well, maybe little tiny pieces of lipid here. So things are really packed in and proteins all interact together. So we know that they interact. So the question is, do they functionally interact? So this story today you're going to hear about is the protein that we're interested in, neuronal calcium sensor 1, which is a calcium binding protein. I'll tell more about it, but it is in the cytoplasm. It's a soluble protein, but it binds to many of the proteins that it regulates. And here I show IP3 receptor and the voltage-gated calcium channel. But today we're only going to talk about this part over here. Okay, so here's the outline. So the first is which players in calcium signaling are in focus, and it's going to be the IP3 receptor and neuronal calcium sensor 1. And of course, we have to talk about the IP3 receptor. It's been studied in a whole host of ways, and three important things that we know about it. One, because of the nice cryo-EM that's come into play in the last few years, we know we have a structure of the IP3 receptor, and so we know where the different components are and have can, based on that, make ideas about what different parts are doing. We can stain tissues with antibodies to the IP3 receptor. Here's a slice through the cerebellum. These are cerebellar Purkinje cells that are white because they're stained with the IP3 receptor antibody. And what you can see here is that everywhere there's an IP3 receptor, everywhere there's ER, there's IP3 receptor. So in every part of the cell, even in these thin axon and axonal extensions. And of course, we know that the IP3 receptor is regulated by a variety of things. In this far right panel, we have the IP3 receptor being regulated by calcium. So it's both activated and inhibited by calcium, but we know from work by many of the people probably in this listening to this podcast or this webinar that phosphorylation and a variety of binding proteins also are regulatory for the IP3 receptor. But today we're going to talk about neuronal calcium sensor 1, which is a member of the calcium sensor proteins. It's the most famous of these is calmodulin, which is shown in the middle here. But you notice that NCS1 is over in the far left, and it's the binding curve is all within the range that you would say was found in the cytoplasm in resting conditions. So it's important for maintaining calcium homeostasis. And we know a lot about this protein. I'm going to show you only a little bit here, but we know that it interacts with a bunch of proteins. This D2R is the dopamine receptor. I already showed you it binds to the voltage-gated calcium channel, binds to some potassium channels, IP3 receptors, that's going to be what we're talking about today. And important for regulating calcium homeostasis and nerve excitability. And because it's an important protein, it's been crystallized, and I think of it as sort of a hot dog bun, and there's this hydrophobic section down the middle, and that's where drugs and proteins tend to bind. Okay, so the first things we did was we were interested in the IP3 receptor and NCS1 interaction. And these experiments on this slide will be talking about NCS1 binding actually increasing IP3 receptor activity. So here we show that we're looking at an IP3 receptor in a planar lipid bilayer, and IP3 is added to one side of the bilayer, and you can see that it's not very robust, but there's channel openings. And we know from looking at long periods of recording that the open probability is about 4%. And if we add NCS1 to the same side as the IP3, you can increase the open probability considerably. So here it's got an open probability of about over 20%. NCS1 alone doesn't do the activity. You have to have IP3. So NCS1 binding alone isn't sufficient to open the channel. And if you use a mutant of NCS1 that doesn't bind calcium, you no longer get the effect. So this is all fine and good. This is two proteins in a bilayer isolated, and I already showed you that probably any two proteins can interact together. So the question is, does this happen in an intact cell? And the answer to that is yes. This is these turn out to be MB231 cells, which are a breast cancer cell, but we've done this in a variety of different cell types, where cells are loaded with a calcium indicator dye, and the x-axis is time, and the y-axis is the calcium concentration. And this blue curve here is the calcium increase in calcium after adding UTP used as an extracellular agonist to produce IP3 in the cell. And based on the single channel experiment shown on the left, when you overexpress wild type NCS1, shown in the green curve here, you get an increase in calcium release. And As again, if you use this mutant that doesn't turn on signaling, in fact, it actually outcompetes the wild type NCS1 in the cell, you actually diminish the calcium signaling. And you'll see in a bit that this mutant of NCS1, L89A, doesn't bind to the IP3 receptor and again outcompetes the wild type, and you again diminish the calcium signaling. So this shows that indeed there's binding. This doesn't show there was binding. We've done other experiments I'm not showing right now that the two proteins bind together and there's a functional interaction. And not only is there a functional interaction, but we know exactly where the two proteins bind. And again, I thank the community for sharing all their reagents, because we've obtained many fragments of the IP3 receptor and based from our friends and colleagues. But we now know, based on looking at all these different fragments, that NCS1 binds to this piece right here, which is known as the suppressor domain of the calcium binding site. So we know they bind. And so based on the fact that NCS1, I already showed you, is crystallized, and we're lucky that this suppressor domain of the IP3 receptor has also been crystallized, we are able to dock the two proteins together, and we can identify the exact amino acids that are necessary for this binding. And we know that this is where this L89 is. And we know that this particular, if you mutate this L89A, as I showed you on the previous slide, there's no longer a functional effect, and there's actually no more binding. We also know on the IP3 receptor exactly which amino acids are important. And we made a small peptide that's equivalent to this piece where it is necessary for binding. And we can outcompete the interaction between NCS1 and the IP3 receptor. And in fact, we can do this in intact cells, because we can take this 12-mer and attach it to a TAT so it's membrane permeate, add it to our cell culture and look at the calcium release. And this dark curve, upper curve, is the release that you get in a wild type cell that just has a scrambled peptide. Whereas if you have the peptide that would outcompete the interaction between the full length IP3 receptor and full length NCS1, you now can diminish the release. Again, providing support that we know exactly where these two molecules, two proteins interact. Okay, so that's the end of the first summary. We know that they bind together and they are functionally important. The NCS1 regulates the IP3 receptor. So now we want to turn to the IP3 receptor and its interaction and how it initiates the side effects of chemotherapy. And so we're focused on Paclitaxel, which is an anti-cancer drug that's also neurotoxic. So Paclitaxel is isolated for the Pacific yew tree. This is the tree. This is the molecule Paclitaxel, and it's used to treat solid tumors. So breast cancer, prostate cancer, those are the two that are ovarian cancer. Those are the kinds that are generally treated with Paclitaxel, and it's known to be a microtubule stabilizer, which is how you inhibit the cancer. But we also know that NCS1 has side effects where it causes peripheral neuropathy and chemobrain. And the question is, well, how does that happen? And at this point, I almost always try to do a shout out to any students that are here to keep an open mind about your experiments and to listen to opportunities that come along, because this whole project started by a random phone call from a colleague in chemistry. And this colleague had Googled PC12 cells and Yale, and my name came up. And he called to see if he could get some PC12 cells. Of course, I said, yes, I'd happy to give him some. And his postdoc and my postdoc were going to meet halfway between in one of the coffee shops halfway between and pass off the cells. And I asked the chemistry professor, why does a chemist want living PC12 cells? And it turned out that he was interested in chemical space and was looking for binders to paclitaxel that wasn't related to the microtubule. And he said that he wanted to test that out in cells. And I said, whoa, did you find anything? Well, yes, he did. What is it? I asked. And he said, oh, you've never heard of it. It's sort of unusual. I said, well, I'm going to give you the cells. You have to at least tell me what it is. And he said it was neuronal calcium sensor 1. And unbeknownst to him, our work looking at the regulation between the IP3 receptor and NCS1 had been accepted for publication, but this was in 2006. So this was before things went online instantaneously. So he was quite surprised to know that we had actually, I knew about it and I worked on it. And so based on that interaction, the four of us met, the two professors and the two postdocs, and we decided to design some experiments to see if there was any functional consequence of the paclitaxel binding to NCS1. And you can be sure that since I'm talking about it, the answer is yes. So this is our model, and I'll go ahead and show you the model and then give you the data that supports this. So the idea is that paclitaxel binds to neuronal calcium sensor 1, and then when paclitaxel is bound in NCS1, the three NCS1, paclitaxel, and IP3 receptors are all together, you get a calcium surge. So you get an excess calcium release because you shift the IP3 dependence curve to the left, and this extra calcium turns on calpain, which is a calcium activated protease, and this calpain then chews up NCS1 and a variety of other proteins. And it's this loss of these proteins that leads to calcium degradation and neuronal damage, and that leads to the side effects. Okay, so now let me show you some of the experiments. First, we wanted to see if there's anything related to calcium related that when you have paclitaxel binding to NCS1, and certainly that's what happens. Here, paclitaxel is added to, this is with SHSY5Y cells, which are neuroblastoma cell line. And the wild type cells have these nice calcium oscillations when you add paclitaxel. And we believe that this is because there's a shift to the left in the calcium, in the IP3 dependence curve, which is sufficient to turn on the channel with the resting levels of IP3 that are in the cell. And if you knock down NCS1, you no longer get this effect. So it requires the binding of paclitaxel to NCS1. Now, this is what happens when you just add paclitaxel acutely, but that's not what happens in cells and in people. So you have a chronic paclitaxel addition, and so this is after 6 hours of treatment with paclitaxel, and it doesn't matter, you get the same result whether you go 24, 48 hours. And now in this case, we add extracellular ATP as an agonist to turn on the IP3 production. And in a wild type cell, you get this nice calcium transient. But in the treated cells, you have a diminution of the response, just as you would expect from our earlier experiments showing that when you lose NCS1, you decrease the calcium signal. Okay, so what about the next step in our model? So we now are saying that we have this excess calcium that then turns on calpain. So in this experiment, again, we add ATP. This is essentially the same experiment you saw previously, where you have the wild type is in this solid curve, and the paclitaxel treated cells are this dotted curve. But now when we add calpistatin, which is a natural antagonist for calpain, we can prevent the loss of the calcium signaling. And we've done this with other chemical inhibitors of calpain, and that we get the same result. So we believe that this shows that not only when you add Taxol or Paclitaxel, you get an increased calcium signal, but this then turns on the degradation of NCS1 and causes a loss of calcium signaling. And here we add thapsigargan to release calcium from the stores to show that indeed we could have released calcium to the same degree in these cells. So the model is Paclitaxel binds to NCS1, and these two then bind to the IP3 receptor. And when you have Paclitaxel, NCS1, and IP3 receptor, you get excess calcium release, turns on calpain, and that calpain then degrades a number of proteins, including NCS1. Okay, so this is fine. It tells you in the cell, but it doesn't tell you that it's having an effect on peripheral neuropathy. So we've done a variety of different experiments. Here we're showing we got, thanks to Professor Pongs, who developed an NCS1 knockout mouse. He generously provided these to us, and we did a Roto-Rod test. And a Roto-Rod test is basically a log roll. You put a mouse on a accelerating rod and to see if they can stay on this rod. And it's a mixed assay, so it has motor, sensory, and cognitive components to it. And so these are wild type and knockout mice. All have been treated with Paclitaxel. And the Paclitaxel, so here's baseline is here, and then you treat them, you give them injections of Paclitaxel to the level that you would expect in a patient. And the blue curve here is the wild type mice, and you can see that they fall off the Roto-Rod after the treatment, and they don't improve with time. Whereas the knockout mouse can not only stay on the rotating rod, but it gets better with time. And this curve, this red curve, which is the knockout mouse, looks exactly like a untreated wild type mouse. I don't show that because it gets there gets to be too many curves, but basically this shows that indeed you need the NCS1 for the Paclitaxel to have the effect on the nerve. Okay, so here's my second summary, and where we have data showing that Paclitaxel indeed does bind to NCS1, and we've been able to show this binding with phage display, NMR, and biochemistry. Then the NCS1 interaction with the IP3 receptor enhances the calcium release, turns on the calpain, degrades the proteins, and causes the damage. Okay, so the next step is, can we prevent these? So we know things about the interaction between the NCS1 and the IP3 receptor. We think that based on that knowledge, we should be able to figure out a way to prevent this side effect. Okay, so the strategies. One could be, the first one would be to block Paclitaxel binding to NCS1. Another is to reduce NCS1 binding to the IP3 receptor. You could rescue the calcium signaling, or you could block activation of calpain. And one of the most obvious ways would be to use calpain antagonists, because many of these reagents were developed to prevent neurodegeneration in the neurodegenerative diseases like Alzheimer's. But it turns out that there are no specific calpain antagonists. Maybe right now there's some being developed, but you need calpain in cells. And so the side effects of calpain antagonism is worse than the actual disease. So even if we as biologists thought this was a great idea, no pharmaceutical company will develop a calpain antagonist, at least not now. So we went for the idea of the NCS1 IP3 receptor interaction to see if we could find a way to modulate this. And in fact, because we had studied this interaction before we knew anything about Taxol, we had some ideas about inhibitors, and that's shown here. And as Aldebaran mentioned at the beginning, my PhD work was looking at lithium transport. And again, a shout out to students that I have often been surprised about how things that I thought would never come back in my life have come back and have become important, because you learn these things and they come back. So the idea was, could lithium, in fact, because we know it has an effect on this NCS1 IP3 receptor interaction, which I will show you in a bit, would that in fact prevent the calcium surge and maintain the neuronal state? And just because lithium has so many interesting components to it, I want to show this. And many people don't realize that 7-Up was originally a lithiated soda, and that 7 comes from the molecular mass of lithium, and up comes because it is a mood elevator. And I don't know if you can read it, but it says, it blends out the harsh features, dispels hangovers, takes the ouch out of grouch. So just like Coca-Cola doesn't have cocaine anymore, 7-Up doesn't have lithium anymore, but it was definitely originally developed as a lithiated soda for mood elevation. Okay, so here's the data. We have IP3 receptor channels in the planar lipid bilayer. That's like I showed you earlier on. And you see that this is the open probability versus lithium concentration. And we have this low IP3 receptor activity just without NCS1. We can increase the IP3 receptor activity when we add NCS1. But as we increase the lithium concentration, we can inhibit, or we modulate, the interaction of the NCS1 so that it doesn't ever inhibit the IP3 receptor to zero. It just removes the activation that's, or the modulation that's provided by NCS1. And for those of you who know that lithium is used for bipolar disease, this is in the concentration range where it's used for treating bipolar disease. So it's clinically relevant concentrations. Okay, so this is in the planar lipid bilayer. Of course, we want to see if there's effects in cells. So in this particular case, we used neurons, so dorsal root ganglion neurons that have been isolated from Paclitaxel-treated mice. Of course, we've done this in SHSY5Y cells and MB231 cells, but I thought that nice to show that neurons, these are primary neurons isolated from Paclitaxel-treated mice. And again, when you add ATP, the solid black curve is the wild type treatment. The dotted gray curve is the mice treated with just lithium. The solid red curve, you see there's a diminution in calcium signaling in the Paclitaxel treated mice, and the mice that were pre-treated with lithium no longer have a diminution in the peak. There is some loss of the calcium area under the curve, but we think that this is it varies from experiment to experiment. So basically, in an intact mouse, we can prevent the loss of calcium signaling. Okay, so that's fine in cells, but the question is, we really want to see if we have an effect on the neuropathy. So again, now we have a mouse study looking at 4 different groups. We have the fully controlled lithium only, Paclitaxel only, and the lithium pretreatment with Paclitaxel. And this is what we used. Again, it's the Roto-Rod test. So R is for Roto-Rod. We have a baseline. We inject either lithium chloride or saline, and we have Paclitaxel or vehicle. And every week we look at their behavior on the Roto-Rod. And these are the data showing the mice. This is the Paclitaxel only, where they fall off the rotating rod too quickly, or they fall off, and they learn, but not as robustly. Whereas if you pre-treat with paclitaxel, these look like the wild type mice. These mice, and they also look like the NCS1 knockout mice. So that we believe that the pre-treatment of lithium prevents the paclitaxel inhibition of nerve. And we've looked at this at a variety of levels, so we can also look at the histology of the peripheral nerves. And I'm not showing the wild type of the vehically, but the lithium only, this is the nerve bundle near where the dorsal root ganglia cells are. And this looks pretty normal, but you can see in the paclitaxel treated nerve bundle, the cells look terrible. There's vacuoles, and what you don't really see well is that the unmyelinated fibers are diminished, whereas if you pre-treat with lithium, the integrity of the nerves look better and the number of unmyelinated nerves is maintained. We do have some preliminary clinical evidence from a retrospective chart review, and I'm going to say that I was very naive. This seemed like a very simple idea. This study with 114 patients from the West Haven VA took 2 years. It was much more complicated, much more challenging than I thought, but we found that there were 110 patients who were provided Taxol for treatment, and this is, since it's the VA, it's mostly men with prostate cancer, and approximately 40% of these people had peripheral neuropathy. And this is the Bayesian analysis shows that this is the curve for the patients without mood stabilizer. We found 4 patients who were bipolar, had been treated, also then got cancer, and none of them have peripheral neuropathy. So it's a limited data, but it's supportive of our hypothesis. Okay, so we feel that it's pretty strong evidence that there's effects of, and we think we know why you get peripheral neuropathy from treatment with paclitaxel, and we think that we can prevent it by pre-treatment with lithium. The question is, can the same effects happen in the central nervous system? And the question is, can we explain why you get chemo brain, and can we prevent it? So we did a meta-analysis, and first of all, so what is chemo brain? So most chemotherapy patients, over 80%, experience some cognitive impairment. They have slowed thinking. They describe they have decreased concentration, difficulty with complex tasks, often, for example, scheduling. So it's considered many of the normal tasks of daily living. And the problem with these chemotherapy induced changes in cognitive function is that most of the cognitive tests are developed to monitor very severe changes in cognitive impairment, like what you would see with Alzheimer's. So that a high functioning individual who would take the test for cognitive function that's developed for an Alzheimer's patient wouldn't necessarily show a robust change. Nonetheless, we found 11 studies published from 1980 to 2019 with over 1,000 breast cancer patients who had received chemotherapy and that were assessed 6 months after they had finished treatment. And we found that there were, in fact, significant differences in attention. There was an increase in depression, and visual spatial memory was altered. So there's robust, or I would call this the best we can do for looking at cognitive function in a human population at this time. But we can look at the mouse and see what happens with the mouse, and there it's much easier to make additional measurements. And here we used open field exploration and the displaced object recognition as a cognitive measurement. Okay, so what is the displaced object recognition test? So here we put mice in a cage and they have 10 minutes to explore the cage. We take them out for 24 hours, and then we put them back in the same cage with 2 objects. And they're there for 5 minutes. Then we take them out for 2 hours. So this would be called a short-term memory test. And we move one object. So there's the displaced, called the D, and the familiar. So, and a normal mouse will spend a lot more time with the displaced than with the familiar, because it remembers that, oh yeah, that I saw that before. What's this? And these are the results. So we're comparing the solid is the displaced object, and the modeled is the familiar object. And you can see that wild type mouse with control injections, they spend more time with the displaced than with the familiar, whereas the Paclitaxel treated mouse does not. They don't remember, and this is measured 23 days post injection. So this is 23 days after their last injection of Paclitaxel. They still don't their memory hasn't returned. However, if we treat with lithium alone, but more importantly, if you pre-treat with lithium before Paclitaxel, we can prevent this loss of memory. So they still remember which is the displaced object and which is the familiar object. And just to make sure, we looked at the total interaction time, and there's no difference among any of the groups, so that the mice are still walking around, they're still curious, but they just don't remember which object was old and which one was new. Of course, with a mouse, you can also look at other components, and we found that the Paclitaxel treatment will alter the axonal tree, the dendritic tree. And so we looked at, so these are the images of the Golgi stained region of the brain. This is in the prefrontal cortex, so part that's important in memory. And if we can look at the basal dendrites first. You see there's no difference among the different groups in the basal dendrites, but the apical dendrites, using the same color code as before, this orange, we have a loss in the complexity of the, or the number of intersections using Scholl analysis of the apical dendrites, but the lithium treated mice have ones that are maintained like the wild type. So that was good morphological evidence. And in fact, not only is it that the dendritic arbor is altered, but the spine density is altered. So here's the saline, this is a control compared to the lithium only. Then if you can see that there's fewer spines in these Paclitaxel dendrites, but if you pre-treat with lithium, the Paclitaxel and Paclitaxel, you don't see this diminution. And in fact, these are images, but the quantitation over quite a few dendrites is shown here, and you get a diminution in the spine density, which is maintained in the lithium pre-treated mice. And just some newer data, we've looked at the hippocampus. So in the Paclitaxel, we looked at hippocampus and prefrontal cortex. We've done the same here for just in the NCS1 knockout. And in fact, indeed, we see a diminution. So this is showing the Scholl analysis. This is the wild type. This is the knockout. You can see that there are fewer dendrites, and it's quantified here where the NCS1 knockout mice have a decreased dendritic complexity. And I can say that these mice also have impaired memory acquisition. So we believe that the model that we prevent for the peripheral neuropathy is also valid for the central nervous system. Okay, so this is our third summary, which is this: we think we can prevent the side effects where the chemotherapy-induced changes in function can be prevented by drugs that alter the NCS1 dependent pathway. And we think that low-dose lithium is a good way to go that way. We know that paclitaxel binding to NCS1 is important for initiating the downstream effects of that lead to neuropathy. And we think that, and we show that lithium decreases the effect of paclitaxel on NCS1 functions, and it maintains intracellular signaling, and it also maintains both sensory and cognitive function. Okay, but this is all fine and good, but if by using a treatment to prevent the side effects, we alter the ability to shrink the tumors, then we have a problem. And in fact, we have been able to show that lithium does not interfere with the efficacy of paclitaxel in breast and lung cancer xenografts. So we did experiments with lung cancer, and we, of course, did looked at tumor volume, but here we're showing the histology that the lithium alone didn't make the tumors grow larger, and it didn't make any effect on the ability to shrink the tumors. And over here, Charles River did the exact same experiments in MB231s, which are breast cancer cells. And what you do is you give the mice, you inject the tumor cells into the mice, and you wait for the tumor to develop for a week. Then the untreated mice are this black curve, and when the tumor gets large enough, in 30 days, you have to sacrifice them because the tumor gets too large. But if you treat with paclitaxel, with or without lithium, you still are able to diminish the tumor size. So the lithium had no effect on the ability to shrink the tumors. So we're good to go here. Okay, so that's the conclusions for the talk. And what I wanted to tell you is that NCS1 is a component of several signaling pathways and modulates calcium homeostasis. We know that chronic administration of paclitaxel decreases NCS1 and attenuates calcium signaling in neurons, leading to neuropathy, both peripheral neuropathy and chemobrain. And we found that lithium protects the calcium signaling and prevents both peripheral neuropathy and chemobrain. Okay, so what's next? Well, of course, the very first thing is that I have to alter my table here and put lithium back into the periodic table because we think that it needs to be it's important. And then we have, of course, a number of questions. One of them is, we don't know what the precise molecular mechanisms that cause the peripheral neuropathy and chemobrain. We're now looking at downstream of the calpain to see what else is affected to lead to actually the loss of nerve function. So we also think that we have a starting point for drug design to prevent other forms of neuropathy, or we would like to hope that's true, because we think that there's a lot of what's called idiopathic neuropathy that we would like to be able to address. And what I showed you is, the story I showed you today is all looking at what happens when NCS1 levels are diminished, but we also know that elevated high levels of NCS1 are also linked to pathologies. And so the question is this also a starting point for drugs for these other pathologies? And you can think of it in the same way you think of, for example, testosterone levels, where too high levels and too low levels are both bad. You need to be in the right window to keep the physiology correct. And so NCS1 looks to be in that same model. And I can say that the clinical trial in cancer patients is now in the planning stages. And this is the team. Of course, this is a project that's been developing since early 2000s. So there's a long list of people who have been in the lab and who have participated in these studies. And they're, as I mentioned, as I went along, the studies are continuing. So at this point, I'll stop and I'll address questions. And I have.
Okay, let me get back on here.
Should I stop? Should I stop my screen so I can see people, or how's that work?
You can keep your screen up because I need to comment on more of Barbara's creations, which the knitting is another one of her skills that not everybody knows about. And you can see her calcium mittens here. So I'll take the liberty of asking you the first question. Yeah, there are many questions. So, and that was a fantastic talk. I was wondering, I was fascinated by your presentation that the dopamine receptor, D2R, is also binding NCS1. Is that interaction inhibited by lithium as well?
We have not done that. I can tell you we know that it binds to NCS1 in a different location than the IP3 receptor, but we haven't looked at it more than that at this point. We have the reagents, we just haven't gotten to that point.
Okay. All right. So we have a lot of questions here. The first is from Taufiq Rahman, and he asks, is the binding mode of paclitaxel to NCS1 known? If so, is it on the opposite end of the side that interacts with the IP3 receptor suppressor domain?
So we think it binds, we think that the paclitaxel binds to NCS1, and we believe it binds to a location close to where the IP3 receptor binds.
Okay.
So in that same hydrophobic pocket, in the same region, not where the dopamine receptor binds, but where the IP3 receptor binds.
Okay. Then we have a question from Carlos Villalobos. Does paclitaxel activate the unfolded protein response?
Ooh, I know that it causes mitochondrial calcium overload, but I don't know about the unfolded. I don't know the answer to that. I'll look into that. I don't think so, but I don't know.
All right. Kevin Foskett asks, how does lithium directly inhibit the IP3 receptor? Is it with the channel? Is it competitive with calcium?
So as far as we know, it's not competitive with calcium. And the answer exactly how it works, we don't know. We tried looking with NMR. We can't, lithium's too small. We can't see where it binds to NCS1. We know that it doesn't inhibit the actual binding of NCS1 to the IP3 receptor. So we just, this is one of our struggles that we're still working on, is exactly how it does it, but we don't think, it doesn't affect the calcium response, and it doesn't affect the actual binding. So it has to be something else.
Can I follow up on that for just a second? Are you, Barbara, are you measuring calcium currents? And it could be a competition with calcium for the permeation pathway.
So we looked at calcium. We don't look at, yeah. So we look at the calcium with the calcium indicator dyes, and the amount of lithium is very small.
I was talking about your bilayer experiments.
Bilayer experiments, well, lithium alone, yes, we looked at lithium alone on the IP3 receptor. It has no effect on the ability to activate the channel with IP3 and with calcium. So it's not directly affecting the IP3 receptor. We did that experiment.
Okay.
So we don't think it's competing. We think it's related to the NCS1 itself.
Okay. Sandip is asking, how does the NCS L89 mutant act as a dominant negative if it doesn't bind to the IP3 receptor?
I think it's just that there's a lot more NCS1 around. And we actually haven't looked to see that it's a dominant negative. All I can say is that when it's present, we don't get the calcium release.
Okay. Dan Johnson asks, Paclitaxel also has cardiotoxic effects, although this is normally chronic. Do you think this could also be IP3 related? Have you looked at any of the cardiac tissues in these experiments?
So in fact, we did look at, we have a whole study on paclitaxel in cardiac tissue. And in fact, paclitaxel does seem to increase the spontaneous beating of cells in a dish. And we did echocardiograms on paclitaxel treated mice, and we could show that lithium could prevent the echocardiographic changes. So we haven't focused on that so much because it's not a very frequent side effect in humans, but we think that yes, there is an effect, and we think it's also through NCS1.
Okay, that's quite interesting. Antonio Villalobos is asking, is there any effect of NCS1 binding to the IP3 receptor on calmodulin binding?
So we were worried that there was going to be some compensation, but we don't see any compensation from any of the other calcium binding proteins. So we think that there's no effect on the other calcium binding proteins. We don't see increases in production of the alpha calcium binding proteins, and we don't see alterations in the binding from others. So so far we haven't seen any effects.
Okay. Martin is asking something a little bit more broad than what I had asked about the dopamine receptor. Yes. Are all lithium effects on neurons fully explained by the NCS1 IP3 receptor interaction?
I doubt it.
Actions of lithium be involved?
I'm going to say I would find it hard to believe that all of them would be. Lithium has too many effects. It acts in a variety of, in other places, both. So NCS1 has many binding partners and lithium does also. So I think that we couldn't say it's just one effect. It's hard to actually tease out in a cell all the different effects.
Correct. Okay. Don Gill is asking, you said that lithium does not alter the action of paclitaxel on tumor growth, but is there a link between the action of NCS1 on tumor growth, or is the action of paclitaxel on tumors completely independent of NCS1?
Okay, so the dogma is paclitaxel only acts on microtubule stability. I'm not convinced that that's correct. We do see effects. So I alluded to the fact that we see effects of elevated NCS1, and in cells with elevated, or we'll put it the other way, in tumors. So we've looked in tumor samples from humans, and we found that the worst prognosis have the highest NCS1 levels. So we found that NCS1 does have effects on mobility and invasion of tumor cells, but it's not clear that that's related to microtubules or Taxol. Does that answer the question?
Yeah, so there is a possibility that NCS1 could increase the availability of paclitaxel and get it to its site of working on the microtubules. I mean, it could assist. It could have some effect on the efficacy of paclitaxel.
So we don't think that that's the case, because in a cuvette, when we have tubulin and paclitaxel with and without, we did that with lithium. Now, did we do that with NCS1? Okay, so I'm going to have to go back. I know we did it with lithium. I'm not sure if we actually looked if NCS1 was involved in the delivery of paclitaxel to microtubules.
Yeah, that would be interesting.
In fact, I would have thought I was worried the other way. I was afraid that paclitaxel binding to NCS1 would diminish the effectiveness of paclitaxel rather than enhance it, and that doesn't seem to be the case.
Okay, good.
Wow, we have still a lot of questions to get through. Here's one from Garrett. It's a three-part question. So the first part is paclitaxel-induced recruitment of NCS1 binding to IP3 receptors dependent on the calcium binding sites of NCS1, and thus calcium binding to NCS1? The second part is NCS1 needed for the paclitaxel-induced effects against breast cancer? I realize your lithium experiment in xenograft appears to indeed argue against this. If not mistaken, paclitaxel has been associated with cardiotoxicity. I think we addressed this earlier. Any ideas whether this could be linked to NCS1 IP3 receptor connection, or even potentially with ryanodine receptors?
Okay, so the heart one, I can say we did not see effect of NCS1 directly on ryanodine receptors. So, and we didn't see so we don't think that the paclitaxel NCS1 is related to ryanodine receptor. Then you asked about calcium. Ask this again. Ask the first one again, just to
So is the recruitment of NCS to the IP3 receptor, is that dependent on calcium binding sites of NCS1?
So NCS1 only doesn't bind to the IP3 receptor when the calcium binding sites are mutated, as far as we can tell. And does that change with paclitaxel? We haven't looked at the effect of paclitaxel on the NCS1 mutants yet. That's actually that is actually one of the experiments on the list. Okay, and the next one was.
It was about the cardiac effects. Let's see. Yeah. Oh, no, sorry. Is NCS1 needed for the paclitaxel-induced effects against breast cancer? I realized your lithium experiment in xenografts appeared to indeed argue against this.
We don't think you need NCS1 for paclitaxel to reduce the tumors. You only need paclitaxel. Paclitaxel can shrink tumors without paclitaxel can shrink tumors without NCS1, as far as I know.
Okay, Martin, how long do we have for questions? Do we at what point do we have to stop?
As long as Barbara can bear it. As soon as Barbara,
okay.
Yeah, as soon as Barbara says she's out of there, then we'll let her go, of course. But it's very kind of her to stay
Good,
with.
because we have a few more interesting questions here. Here's one from Colin Taylor. If the critical step is a loss of NCS1 evoked by an IP3 evoked calcium signal, what's so special about a paclitaxel NCS1 evoked calcium signal? There seems to be a feedback loop whereby NCS paclitaxel gives the calcium signal that activates calpain and degrades NCS. So how is anything sustained? Surely once NCS is degraded, there is no calcium signal, no calpain activation, and NCS should recover.
So this is one of the things that drives me crazy, because the paclitaxel in a person is given once a week, and paclitaxel isn't around all the time. So the question is, why is it that, and you would think that the NCS1 levels would recover. So the answer is, we don't know yet. This is something we're actively working on. I have a variety of ideas, which Colin, you and I can talk about offline, but so the measurement says that the NCS1s doesn't recover, and we know that it's not just NCS1 that is degraded. There are quite a few calpain targets in the cell that are related to neurodegeneration. So we believe that there are additional components that are degraded. We use NCS1 as a surrogate to show that the paclitaxel is having its action, but we believe that that's not the be all and end all. We think I was trying to be careful that it initiates the whole process. We don't think it's the end. That's why we're interested in the mechanism for the neurodegeneration. Does that make it clear?
Clear to me. Natalia Prevoskaya asks, neuropathies or multifactorial pathologies? NCS1 IP3 receptor is only one of the pathways involved?
Yes. We like I just said, we think that, well, one of the things I like about this particular neuropathy is we know exactly when it starts, and so we can look at the initiation. Once you initiate the whole downstream path, we think that there's a variety of ways you can go. And that's of course the next step in this whole process. We can say that it happens, we can prevent it from happening, but and we're happy we're fairly upstream, but we know there's a lot more to learn about downstream.
All right. Taufiq Rahman gives many thanks for the excellent talk. And Alex Simpson says, enjoyed the presentation. Does paclitaxel bind other NCS1 family members?
So that I can answer. When we did the phage display, we looked at all the different calcium binding. I didn't do it, the chemist did it, and we didn't see anything. It was very specific for this particular calcium binding protein. So we think it's reasonably specific.
Okay.
Okay, thank you.
And then Garrett has a follow-up. Would the effect of lithium depend on the calcium binding site of NCS1?
So we haven't mutated each of the individual calcium binding sites. We've only done one so far. So I can't, so the real answer, I can't give him the answer, the specific answer he's going to want. But that's something that we'd have to look at, that if the different, because there's NCS1 has like calmodulin has 4 calcium binding sites, it binds 3 calciums, and we haven't looked at each individual one, so that would have to be done.
Thank you. The question arose when you were discussing with Kevin Foskett in that condition. That's why that question was connected to that. Yeah, thanks a lot.
Okay. Amalia Dolga says, thank you for the clear talk. And then she asks a great question. Does paclitaxel alone or in pathology affect the ER mitochondrial contact points?
Yeah, so we haven't looked at that. That's a very good question. That's something that's we do, and we could do that, but we haven't yet. Good question.
All right. Then we've got, let's see, some thank yous from different people. Antonia Villalobos for the very innovative work, and Dan Johnson, sorry. Jonathan Sobolov asks, does the lack of competition between NCS1 and microtubules for taxol binding reflect higher affinity for microtubules?
Turns out that the affinity is similar. It's maybe slightly more for microtubules, but it's not an order of magnitude difference. So my guess is just there's so much taxol around that it doesn't matter in a person that's getting treated, but something that could be tested.
Okay. And from here on out, we have many compliments about your nice talk, fantastic talk. Great to see you. Thanks, Barbara. Great to see you again from Colin, Jonathan, many other people. So with that, I think we can just open up for casual discussion. Is that right, Martin?